Hello, everyone, and welcome to our final broadcast of this three-part webinar series that is being put on by the ACS Division of Polymer Chemistry. I'm Mike David, and I will be your host for today's broadcast, which will focus on polymer chemistry in biomedical applications. We will be joined by Andrea Casco, an associate professor in the bioengineering department at UCLA, as well as Nathan Janeski, the Jacob and Rosaline Cohn Professor of Chemistry, Material Science and Engineering, and Biomedical Engineering at Northwestern University. Our moderator today is Mark Hillmeyer, who is the chair of the Division of Polymer Chemistry, as well as the McKnight Presidential Endowed Chair at the University of Minnesota. If you have any questions or comments, you can enter those into the questions panel. The slides for today's webinar can be downloaded from the control panel, and the recording will be sent to you once it has been posted. And now, I'm pleased to turn the time over to Mark.
Thanks very much, Mike. I'm very happy to be hosting this third in the 2017 series of webinars hosted by the Division of Polymer Chemistry and ACS Webinars. We thank them for all of their support throughout this series. And it's been my honor to be the chair of the Division of Polymer Chemistry for this 2017. And I've been happy to be hosting these seminars and bring content from outstanding scientists from across the nation to folks in this electronic format. Most of our members, in fact, are unable to attend the national ACS meetings. And I have a long running streak now, attending many of them in a row, where I get to see talks like you're going to hear today. So the Division of Polymer Chemistry really does want to bring value to its membership that are unable to attend ACS national meetings. So we try to pick contemporary, interesting topics from experts in the field, and we're very happy to have Professors Casco and Janeski here today with us. And so without further ado, I'm going to turn it over to Andrea to get us started here. But just remember, if we do have questions, you can use the question box to ask those questions during Andrea's presentation or Nathan's presentation. I'll be collecting the questions, and then we'll go both presentations in a row, and then at the end of the presentations, I will moderate the question and answer session for both speakers.
Thanks, Mark. Let's jump right in. So generally speaking, there are four main ways that polymeric materials are used in biomedicine. They're often used as structural replacements, for example, as implants, in drug delivery, and you'll hear more about that later today with Professor Janeski's talk, as scaffolds for tissue regeneration, which I'll be talking about, and in devices and diagnostics to monitor health and disease detection. So let's look at a commonly used polymer in biomedicine, silicone, and see how it's used in each of these examples. So we're all familiar with breast implants, which are shown in this example here, and they're made of silicone and often filled of silicone. In drug delivery, this example is Norplant, where you have silicone tubes that are filled with hormones for contraceptive purposes, and they're implanted under the arm, as you can see in this picture here. Silicone is also used as a scaffold. In this case, this is a commercial product called Integra, which is used for regenerating skin after diabetic foot ulcers and other wounds to the skin. And you have a semi-permeable silicone membrane on the outside of this particular construct. And then in this last example, Google a couple years ago reported these smart contact lenses, and contact lenses are typically silicone hydrogels. And in this case, it's a diagnostic device that monitors the glucose level of diabetic patients and alerts them to when they may need to take insulin. So these examples illustrate how polymers are generally used in biomedical applications. But as polymer chemists or polymer scientists, we obviously have access to more polymers than just silicone. This leads us to our first challenge question.
Our first question that we'd like to ask you is, besides silicone, which polymers are commonly used in these biomedical applications? Well, Andrea, our number one was polylactic co-glycolic acid with 85%, and the next highest was polyethylene with 45%.
So in this case, all the answers are correct except perhaps polystyrene. All of these polymers are used as biomedical implants. Polystyrene is typically not implanted in the body, but is often used in biomedical applications outside the body. So moving on, when we think about designing polymeric materials to interface with living systems, we can take inspiration from nature. In nature, cells live in the extracellular matrix, which is basically a hydrogel that is interspersed with polymeric fibers, typically collagen. A lot of implants are composed out of solid polymers, and these might be used as flat surfaces for cell culture, as fibers, as 3D printed structures, or as you can see on the right-hand side of the screen here, these are Teflon vascular grafts. Many researchers also focus on hydrogels, which are cross-linked water-swollen networks that allow both two-dimensional and three-dimensional culture of cells. And these are often mechanically defined, topographically patternable, and you can make them degradable. And hydrogels is what my group focuses on and what I'll be talking about today. So in this slide, I've listed numerous challenges in the field when it comes to making hydrogel scaffolds for growing cells. And I've underlined three in particular that are important. First of all, when it comes to nature and developmental biology, chemical and physical gradients are incredibly important, and yet these are really difficult to recapitulate biomaterial, particularly on small scales down to the micron level. If we look to nature, we also know that signaling cascades are extremely important, so we have to be able to deliver cues in a temporally controlled manner and to dynamically remodel our environments. And then from a practical point or a practical perspective, scalability is an issue. We can make lots of really cool biomaterials in the lab, but often if we think about translating them into the clinic, it's just not feasible. So today I'll be talking about some of our efforts towards addressing these underlying challenges using hydrogels. And we can begin with the question, how can we build a dynamically tunable biomaterial? And more specifically, since my group works on hydrogels, how can we make a dynamically tunable hydrogel? The way we do this in my group is using light, and more specifically via photodegradation. Light is ideal because you can easily turn it on or off, and you can focus it spatially so that you only have your reactions at a specific location. So this gives us both temporal and spatial control over degradation and therefore over material properties. If we imagine a hydrogel that contains photodegradable linkages, there are a couple of different ways we can use them. On the left-hand side over here, you see we have a photodegradable group that will tether a therapeutic agent into the hydrogel, which is represented by this disc. The alternative way we can do this, or a different way we can use this, is to take a photodegradable group and incorporate it into the backbone of the hydrogel network. And this will give us control over the mechanical properties of the hydrogel. So I'm going to talk about some of our work in which we incorporate a photodegradable group into the backbone of the network. And in our case, the blue photodegradable group is an orthonitrobenzyl group, or ONB, as I'll refer to it throughout this presentation today. And ONB groups are ideal because they photodegrade at cell-compatible wavelengths and under cell-compatible conditions, and they've been used as small molecules as photocages for neurotransmitters applied to live cells. So that implies a certain amount of biocompatibility. The mechanism of their photodegradation is also well characterized so that we understand what products we get in the system at any given point in time. We can use our knowledge of basic polymer science to predict how degrading linkages in the backbone of a photodegradable hydrogel network, or changing the crosslink density, which is going to be a function of the degradation, will affect the macroscopic physical properties of the hydrogel. So we know that the swelling ratio, the volumetric swelling ratio, will increase with exposure time in a predictable way. The elastic modulus of the hydrogel will decrease with increasing exposure time. And the diffusion of small molecules throughout the gel will also increase as a function of exposure time or decreasing crosslink density. We've published quite extensively on the area of photodegradable hydrogels, so I'm going to highlight one of our recent papers here that addresses the issue potentially of scalability. So we recently reported using a maskless lithography system for rapid patterning of photodegradable hydrogels. In this system, a UV light source with a bandpass filter illuminates a digital micromirror device. And so you input a grayscale image into the system, it gets inverted, and the image pixel intensity values get transcoded by the digital micromirror device into a grayscale pattern. So this is an input image that we chose, Starry Night by Vincent van Gogh. It's quite recognizable. And then you get the negative image that's projected onto the hydrogel by this digital micromirror device as it passes through a reduction lens and gives you a collimated UV light pattern. The resultant hydrogel, you can see we get faithful reproduction of the Starry Night image on the hydrogel. We've actually fully characterized this by AFM and other characterization techniques, and we can actually see quite good resolution in this hydrogel. And what's nice about this, you'll notice that the scale bar here is 100 microns. We can actually, simply by adjusting the instrument parameters, now make an image that's now 1,000 micron scale bar or one that's 50 microns. So this gives us the ability to easily change the image or to easily change the the scale. You simply adjust the settings or upload a new image file into the system. So it's a way of rapidly producing patterns on hydrogels without having to reprint photomasks, realign the system, or anything else like that. So something that's a little more biologically relevant than Starry Night, we wanted to use the system to produce mechanical gradients in our samples to see if cells had any sort of preference for a specific substrate. So we used the gradient image that's shown in A over here, which is a linear gradient of pixel intensities, and we repeated it multiple times across a hydrogel in order to generate a number of replicates shown there. We have two different compositions of hydrogel, a 10 weight % hydrogel and a 20 weight % hydrogel, just to give us different stiffness ranges. And we measured the stiffness of the hydrogel or the modulus using AFM as a function of position along this gradient. And what you can see is that we have a monotonic decrease in the gradient as a function of exposure time. And we have a nice range here of anywhere from up to 220 kilopascals in the 20 weight % gel down to about 10 kilopascals in the 10 weight % gel. And these values are important because this is a physiologically relevant range of moduli that is often used to explore the effect of material stiffness on cell fate. We wanted to determine what the resolution was of our system, so we actually used a photomask file of a bunch of different lines of decreasing line width. So we projected this image onto our hydrogel and then used AFM to detect the lines or characterize the topography of the system. And the smallest width line that we could detect was about 850 nanometers, as shown in this graph here. So we've got submicron resolution and a continuous gradient capabilities within the system. We wanted to test to see how cells would respond to this system, so we seeded human mesenchymal stem cells onto these gels. And I should add that we added a cell adhesive peptide to these gels so that the cells would stick. And to our surprise, so this is the projected image, the negative, and then this is the resultant hydrogel. This is an AFM image of the topography where you can see in the areas that are not degraded, we have valleys, and then the gel swells up and we get these softer regions that project outwards from the surface. And when we seeded the cells onto these gels, the cells aligned preferentially onto the softest part of the gel that also had the highest topography. And this was a little bit unexpected because in the literature, it's often stated that cells prefer stiffer substrates and they'll often fall into the valleys in patterned substrates. So we wanted to test how robust this particular effect was. So we wanted to test how robust this preference was of the cells for these soft and higher topography regions. So we decided to throw them a bit of a curve. And because we can rapidly pattern our gels simply by uploading a new image file, this was something we could do in the matter of hours instead of days or weeks. And so we used the same gradient pattern. We patterned it in two different scales shown on the left here and the right here. And then we added curves into this pattern to see if the cells would continue to follow that particular modulus or if that preference would break down. And as you can see from the micrograph here, the cells have a pretty strong preference for that particular modulus in this system. So you can see both for the larger scale gradient and the smaller scale gradient that the cells do quite faithfully follow those curves. The only thing that we see is that when the pattern is small enough, sometimes we'll see that the cells will jump over one curve and onto the other. You know, it was really interesting results, but ultimately the beauty of this maskless lithography system was that we could rapidly design and test a series of different patterns, including these ones of different size scales and in these different curves. And we had many replicates on a single substrate. And these experiments would have taken significantly longer if we had used just simple photomasks instead. So now that I've demonstrated how light can be used to rapidly generate novel hydrogels for culturing cells, I pose the next challenge question to the audience.
Our next challenge question is, what other stimuli have been used to dynamically control polymetric biomaterial properties? You know, these are all pretty much even, well, except for magnetic fields got 77%, but none of them got below 60.
All of those stimuli have been reported in the literature. I would say the most commonly reported one is actually using enzymatic degradation in order to control material properties. But there are examples, particularly in drug delivery, for magnetic field ultrasound and mechanical force as well. So now I want to move on to a new story, but we're still going to be using the same photodegradable hydrogels. And in this one, I'll be talking about shape-changing hydrogels with the goal towards replicating physical transformations in developmental biology. So in this case, we started with a very simple question. How do we, as human beings, go from a homogeneous mass of cells, seen here as the inner cell mass, to the fully formed human being that we see here? So in this case, this is a UCLA gymnast, Kyla Ross, who also happens to be a student in our department and a former Olympian. And the key step we were interested in this process is really capturing an event that occurs after gastrulation. So during gastrulation, we form three germ layers, the endoderm, the ectoderm, and the mesoderm, shown here on the right. And these are three flat sheets of cells. And these three flat sheets of cells undergo a series of folding transformations that ultimately lead to nearly all tissues in the body. So for example, we have two flat sheets of cells that fold into tubes, and those two tubes fuse together, and that forms the primitive four chambers of our beating hearts. So we wanted to ask the question then, can we use photodegradation to generate folding in hydrogels? And so one of the properties of photodegradable hydrogels is that you naturally generate a gradient due to the absorptivity of the degrading species and the degraded species. And we've characterized or quantified the molar absorptivity of our photodegradable orthonitrobenzyl groups, as well as the degradation products at multiple different wavelengths, as you can see on the table here on the left. What this means is if we shine light onto one side of the material, we have a region where the crosslinks are degraded. We then generate a gradient, and then we have another region where they are intact. So this means that we have differential swelling from one side of the hydrogel to another, and that can generate a bending moment within the hydrogel. So our approach is outlined here, where we take a hydrogel, we expose it to light from one side. That UV light is attenuated through the gel thickness that results in a crosslink density gradient. So we have regions of lower crosslink density on the surface closest to the light source and regions of higher crosslink density on the non-exposed side. And when we place this in an aqueous system to equilibrate, it should naturally fold due to the differential swelling that occurs. So the question is, does this work very well? So we took different hydrogels of different shapes. So we have circular hydrogels and square hydrogels, triangular, and then long, thin strips. And we exposed them to UV light under a number of different conditions. For the circular hydrogels, as well as for the squares, we just expose one side with a flood exposure. And these will fold into tubes. So you can see the tube formation end on and side on in this particular graphic. If we mask the center of this triangle here and only expose the edges from one side, it will fold up into a little container. And if we expose the long strips of hydrogels using a striped pattern shown here, then what we do is, what we observe is spiral formation. And this can be seen a little bit more clearly in this video. So this is a spiral hydrogel that we've dyed in a blue dye. And what you can see is that basically, there's a preferred gel conformation or shape, in this case a spiral, due to that differential swelling of the hydrogel. And even if you deform it, it returns back to that spiral shape. So in order to use this in a biological application, we need to be able to do this in the presence of cells. And there are two different ways we envision doing this. The first one is where we take our macromers, our photodegradable group, or macromers shown here, and we co-polymerize it with a non-degradable PEG to form a hydrogel sheet. We can then seed cells onto that sheet, expose it to light from the other side, and it should naturally form a tube. The other possible way we can do this is we can take our macromers, mix cells in with those, polymerize it into a sheet so that we have encapsulated cells, expose it to light, and then we should get tube formation. And the difference between these two here is that using the top approach, we have cells that are going to be seated on the interior of this tube, and in the bottom approach, we have cells that are seated within the tube and not within the interior. So we chose to use a particular cell type called C2C12 cells. These are muscle cell precursors. And on the left-hand side, we're showing you the basically inner diameter of the hydrogel tube as a function of the intensity of light that we used as the exposure source. And then in the insets here, you can see the shape of the tubes that form. Now, what's important about this is that these tubes actually, although you can't see it in this picture, actually have cells seated on the interior of these, and the cells have very high viability. We then took these particular tubes, and we stain the cells within them using a red stain, a red fluorescent stain for the cytoskeletal actin, and a green stain for the nuclei. And what you can see is that the cells survive the process, and they even begin to proliferate. If you extend the culture a little bit longer, what we can start to see is that the cells will adopt a more elongated morphology, which is consistent with the differentiation of the C2, C12 cells from the myoblast state to the myotube state. And you can also see that they proliferate and begin to fill the volume of the tube. So in this last slide of data here, we did the experiment where we encapsulated the cells instead of seeded the cells. So the cells are entrapped within the hydrogel in this case, and then we expose them to UV light to generate folding. What we see in these micrographs, these cells are stained with a live dead stain. So the live cells are green and the dead cells are red. And regardless of the exposure, the light exposure intensity, we see about the same amount of live cells and dead cells, including in the sample that wasn't exposed at all. So this indicates to us that the cell viability is really an issue of the encapsulation process and has nothing to do with the UV light. The folded sample is shown in the lower right-hand corner here. Again, with this live dead cell, you can see that there are live cells throughout the entire volume of the hydrogel. In this case, it wasn't completely folded. It only folded into a semi-circle here, but we have been able to since generate fully folded cell encapsulated tubes. So this brings me to the conclusions for this presentation. We've shown that photodegradable macromers containing O and B linkers can be utilized as dynamically tunable biomaterials. Using maskless lithography allows rapid patterning of hydrogels with submicron resolution and the generation of many replicates within a single system. Attenuation of light by the O and B group generates a gradient in degraded hydrogels, and that gradient generation can be used to induce folding in our hydrogels in ways that are compatible with live cells. So this brings me to the end of the presentation. I'd like to highlight the students who performed this work. So Stephanie Delgado and Ellie Capilla were the two students who performed the shape-changing experiments, and then Sam Norris is the student who performed the maskless lithography experiments. And I should acknowledge support from a number of sources, including the NIH, NSF, JSR, and Millipore corporations, as well as some fellowships for my students. And with that, I will turn the presentation over to Nathan.
Today, I'm going to tell you about some of our strategies that we've developed in my lab now at Northwestern University towards trying to target materials and ultimately the drugs that they carry to various tissues. In particular, we're interested in whether or not we can target to diseases of inflammation. And so we've got several examples shown on this slide here of both cancer in the center of the slide here, and to heart disease. Now, our lab is interested in the development of soft materials that, as I mentioned, can carry small molecule drugs and certain types of biomolecular drugs towards these kinds of diseases. Now, these have to be done via intravenous injections. And then, as you can see in the middle, we can target these in various different ways. We've worked a little bit on delivery of agents that you can observe via imaging in the whole animal, as shown in the center. And on the right-hand side, in the case of cancer, and on the right-hand side in the case of heart disease. I'm going to tell you a story about the delivery of cytotoxins in the case of where we want to damage the tissue, and that is we want to kill off tumor tissue in the case of cancer. And on the right-hand side, where we want to actually heal the tissue. We haven't quite got there yet, but what I'm going to describe to you is our efforts towards developing agents that are capable of doing that. We term these enzyme-directed assembly of particle therapeutics. This is the strategy we've developed, and I'll describe how that works over the course of the next few slides. Let's back up a little bit, though, and start at the beginning of this. And where we really got interested was sort of in the fact that there is simply no magic bullet. And so this idea that you can develop a very well-targeted agent that can be intravenously injected and will then go and seek out its target without having any other effect is just not reality. And so our real aim, whether we're developing polymeric carriers, nanoparticle-based carriers, or even small molecule modifications in traditional sort of medicinal chemistry approaches, our real aim is to expand the therapeutic index. And so you can see a plot here of sort of the traditional view of this. This is really the window of opportunity, right? This is the ratio between the dose that gives us a toxic effect in the majority of people or animals or patients and whoever we're treating, and the dose that gives us a toxic effect, and then the dose that gives us a therapeutic effect. The difference between those two gives us a window of opportunity in terms of the safety of the drug. And so ultimately what you want is a very, very high toxic dose and a very, very low effective dose, such that the majority of the population can be dosed safely. What we typically start off with in the case of chemotherapeutic drugs are drugs, small molecules in some cases, that have very narrow therapeutic indices. That is a narrow safety window, so that they're toxic. And we know this from just our common knowledge that chemotherapeutic drugs, and most of these are traditional cytotoxic drugs, make people sick. So the degree to which the patient can tolerate that then determines the dose that they're ultimately given. Again, when we formulate nanoparticles, what we want to do is increase this tolerated dose of the drug that it's carrying. What typically happens then, if we achieve that goal, is that we lock the drug up so well that the effective dose tends to go up. And so you shift this entire window to the right without ultimately changing the safety of the drug. Alternatively, you can develop carrier systems that don't ultimately alter the tolerated dose at all. They don't alter the effective dose, and the carrier system just serves as a way of solubilizing the drug. And there are plenty of examples of both of these ways, in a sense of failing to develop a safer drug or a better targeted drug. These are quite prevalent in sort of the field. And so there's a significant challenge in overcoming the barriers to the basic therapeutic index associated with the parent drug or the small molecule that we aim to deliver in our carrier system. And this leads us to our first challenge question, because this relates to us being able to benchmark our systems to FDA-approved formulations.
Which are commonly classified as nanomaterials. Is it Herceptin, Taxol, Abraxane, or Doxil? Well, the majority of the audience, at 58%, said it was Doxil, and then the next highest was Abraxane.
The audience is on track here, and these are, of course, famous formulations, and you see them on the right-hand side here. These are electron micrographs showing the nanoscale morphology of those particles. And you see on the top right just a beautiful example of a liposomal formulation of doxorubicin, which is the commercially available and approved Doxil formulation. On the bottom right, you see Abraxane, which is a human serum albumin, Taxol kind of composite type particle. Both of these, as most of you correctly said, are FDA-approved formulations. And these are the types of things that we want to benchmark against when we develop new targeted or untargeted carrier technologies. These are approved for various different reasons. And some of these relate to changes in pharmacokinetics that give you less toxicity upon injection. Some of them, and these are included, change the solubility profile and therefore the bioavailability of the drug. So there's lots of different targets, and I've sort of pinpointed the ultimate goal of expanding therapeutic index, but there's lots of different ways of going about this problem. In the center of this is shown sort of just one article that I pulled out. This is a review article, but you see these kinds of parameters crop up all the time. Okay, how big a particle needs to be, maybe what its surface charge needs to be, maybe how big it can be given its shape, and all these different parameters. What we're interested in is the different parameters that are at play that define the pharmacokinetics, how long it stays in the bloodstream, and ultimately where it distributes within the body, how these parameters can be changed in time, not just a static system. So we got interested in how a particle could be made stable in the bloodstream, for example, but very unstable within certain tissues. One way to look at this might be a particle that's injected in one particular charge state, but changes charge once it targets to a location. And by picking on different properties of different tissues, could you get controlled biodistribution through switchable systems that change their tendency to be retained within different tissues at different times. And so our approach was really to look at this idea of dynamics from the standpoint of surfactant assemblies. And many of you may be familiar with this. This is sort of the famous kind of picture on the right-hand side here of how surfactants, in this case drawn as maybe a block copolymer, assemble to give you different morphologies. The major driving forces for this are a hydrophobic portion of a molecule, in this case shown in red, and a hydrophilic portion of a molecule, in this case shown in blue, packing together to mainly for the reason of excluding water. So this is the so-called hydrophobic effect. And so you drive the particle to form by excluding water onto the outside of the particle and throughout the hydrophilic shell, excluding it from the core, which is now hydrophobic in water. And so in this way, you can imagine encapsulating drugs on the internal portion of a spherical micelle, as shown on the top. Now, as the geometry or the geometry at equilibrium of the surfactant changes, the equilibrium structure changes to this more lamellar type architecture, as shown down on the right-hand side as a vesicle. And this is where we imagine the surfactants as packing as cylinders rather than as cones, giving us this sort of flat surface on the nanoscale. Now, in reality, our materials and those that many others generate using these properties, what we see is a range of different morphologies, some of which follow these rules in a straightforward way, and many of which don't. And so we have ways then of pushing particles back and forth across these kinds of energy landscapes where the very same polymers can be shifted between phases. And these types of materials will have very different properties in vivo. Now, if we could have biochemical signals take particles between these different structures very selectively, then we'd have a way to design materials for targeting to the signals that initiate those different changes in phase. This is sort of shown again here on this diagram, where now we have a stimulus that's actually changing the three-dimensional structure of each surfactant. This is shown here in a very, very simple cartoon to sort of stress the point, but you might imagine a simple surfactant, a polar head group and a hydrophobic tail. They pack together closely in order to, again, exclude water from the core. So this is an entropic effect, and give you this kind of spherical micelle. Now, if we have a way of decreasing the size of the polar head group, we could potentially shift these particles from a very condensed, very small spherical micelle to a larger architecture, either a lamellar bilayer, the liposome that I showed you on the previous slide, or some more complex network morphology. And that's exactly what we envisioned, whether or not we could use specific biochemical signals to initiate changes in phase to get changes in shape. So in this slide on the right-hand side is shown one of the very original diagrams that one of my students drew up as we were developing this idea. And the concept was, could we inject a very small particle, a very small polymer, or even a small molecule that's essentially injected as a progelator? So these are materials that would have fairly rapid circulation and clearance times. So they would, and that's not exactly depicted here, and we haven't exactly achieved that, but that was the original goal. So we'd have a material we could intravenously inject safely that would then form a very large microscale particle within the tissue. And so you see that in the cartoon, an intravenous injection, you get circulation of these particles, and they continue to circulate, and potentially they clear through the renal system. They also passively equilibrate into tumor tissue, where they encounter enzymes. Now, these are enzymes that are overexpressed by immune cells associated with the tissue and also by the tumor themselves. These are part of the inflammatory response. We're particularly interested in enzymes that proteolytically destroy the extracellular matrix that surrounds the cells, that builds up the tissues. As this occurs in metastasis, the cells are able to leak into the bloodstream. Our particles respond to those exact same signals by mimicking the extracellular matrix itself. They spring open and they stick to the tissue. These biomarkers were validated and continue to be so as highly active within the tissue by a variety of different groups, including my colleague at UCSD, Roger Chan. And so we took up on these ideas of whether we could use this kind of catalytic biomarker as a way to build up a material, physically entrap it, rather than having the particle stick receptor that's on the tissue, so that you'd have a very strong driving force for accumulation, and therefore get away with this idea of having a very rapid circulation time and clearance. And so this is the basic sort of diagram then of how some of these materials are made, some of the chemistry of this. On the left-hand side here, you see the monomer, right? M equivalents of this monomer, this strained olefin that these ruthenium initiators go right after. M equivalents of the monomer give you M-mers approximately. And this works quite well in this case, a living polymerization method to generate a block that contains the drug as a hydrophobic portion. As I mentioned, lots of these drugs are hydrophobic. We can then add into the mix N equivalents of a peptide. And this is a peptide sequence that derives from the extracellular matrix of the tissue itself. And so we end up with a block copolymer, A, B blocks, which is just a very fancy surfactant, a very expensive, fancy surfactant of the type that I showed you in that red and gray diagram earlier on. The block sizes and the nature of those blocks then define the phase or the morphology of the particles. And shown in this figure here, labeled C, is the initial structure of the spherical micelle that are formed. When we add enzymes, the enzymes and combinations or individual enzymes that are produced by these cancer tissues within the cancer tissue, as I said, that are responsible for cutting free these cells, the particles undergo a phase transition. As the peptide gets depleted, you decrease the size of that polar head group and you get an aggregation event. Now, this is actually a true change in the nature of the particle itself. It exposes the drug and then the drug begins to leak. And so you've tied together the ability for us to kind of form this aggregate or this kind of implantation event following intravenous injection with a drug leakage profile. So how does this actually look? This is the chemistry that we actually use to make these particles. You can see here a strain norbornene ring with our Taxol monomer. When we append it to this 2' hydroxyl, we kill the drug. It's not effective when it's in this state. It's a prodrug type formulation then. We polymerize it, as I said. What we can do is, because it's a living polymerization, we can first polymerize the drug, and then while the ruthenium catalyst is on the end of the polymer, we can add either the L-amino acid sequence, which is recognized by the enzymes, or a D-amino acid version, which is not. It's the control. So it's exactly the same sterics and electronics, but of course the wrong enantiomer in the peptide. And so we have the polymer as shown here, which is a very short sort of oligomer of drug and responsive element. And we have the control system, which isn't shown, which is the D-amino acid sequence, the exact analog. About 100 of these packed together to give us the spherical micelles that are shown in the micrograph. This is a standard negative stain electron micrograph in the background here. So what happens when we go in vivo? And this is a lot of truncated data here just to make this point quickly. This is the type of thing we do initially. So a tolerated dose study. As I said, what we want to have is a very high maximum tolerated dose as a starting point, at least for these types of cytotoxins. And so what we do is we obviously weigh the animals and then we begin our studies. And so you can see here a comparison to the clinical formulation of Paclitaxel. This is quite toxic upon injection, but you can see the mice tolerate 15 milligrams per kilo. That's with respect to the drug. That's a sort of a standard dose of that drug. If you got to 30 milligrams per kilo of that parent formulation, the clinical formulation, you can see that the animals don't tolerate this and they go through the weight loss threshold and are sacrificed. Our formulation, the Paclitaxel carrying responsive particle, is now safe at 30 milligrams and safe out to 240 milligrams. These are very, very high tolerated doses. The real question here then becomes, is it actually effective? You're dosing a lot of drug and you're seeing minimal effect on the animal. Then are you also inhibiting the drug from working against the cancer? And this is kind of the interplay, right? I mentioned as you make a very good encapsulant for the drug, you can also kill off its activity against the tumor. And so the next question is, does it work via our approach? Is it true that this shape change mechanism has an effect? And then ultimately, does it work at these lower doses, the 15 milligrams where Paclitaxel is known to work? Does that mean then we have a safe drug or not? And so as I said, the first step is, does it work via our mechanism? And I mentioned that we made this L-amino acid polymer with a peptide, with the L-amino acid peptide. That's responsive to the enzyme. It acts as a substrate. And then the D-amino acid control, and that's depicted here in red. This is, as I said, a peptide sequence that has a very high Km, a very low affinity for the enzyme as a substrate. These are injected intratumorally into straight into the tumors of the animal to give the control particle as much of an opportunity as possible to actually work. And it doesn't. It completely leaks out. So I should note that these control particles also don't work when they're intravenously injected either. But this is sort of the best example we can give of where this idea of physical entrapment is actually working as this kind of new way of thinking about actively accumulating materials. You can see in blue that these work quite well. And again, this is working at the low dose, but we're cheating. We injected all of it into the tumor. And so we would assume that Paclitaxel would actually work if it were all inside the tumor. And so the next step is to go against, as I said, and as you guys guessed, FDA approved type drugs, or rather than guessed, actually knew potentially, these FDA approved type formulations. In this case, Abraxane and Paclitaxel, not the nanoscale formulation, but the commercial one. Never mind the doses here, just to simplify this, we went down to 15 milligrams per kilo, which is the effective dose for Paclitaxel. And we can see the same outcome. So over 14 days, and this again, just a brief amount of data, and I'll show you some more in a second, we can see that at the low dose, we have efficacy. At the high dose, we have safety. And so we're beginning through, and this is what's really exciting for us, we're beginning through this new approach at thinking about how to accumulate materials to see this kind of concept of this broadening of the therapeutic index and this safety window opening up. Now, this is the type of study that's really ongoing in our group now. This is a slightly different formulation in the 5 milligram, 20 milligram, and 250 milligram doses that I show you in the colors here. But this really illustrates where we really want to be when we head to head against commercial things like Abraxane. You can see here on the right-hand side is a traditional Kaplan-Meier curve or a survival type curve where, of course, 100% of the population is alive at the beginning of the study, and then the animals die off from cancer when they're given saline, or they're killed by the drug if the drug is toxic at that dose. And so you can see now in the formulation that I'm not completely describing what happens when you can get up to very high doses. And here, 250 milligrams per kilo, the entire cohort of animals survives that dose. And you can see the massive difference between saline and even the low dose of the commercial materials. This has a lot of development to go. There's a lot of things that aren't depicted on this plot, including ease of formulation and other things and other considerations. But this is the kind of thing we want out of a targeted therapeutic. And the real question and the real challenge in nanomedicine is, can we indeed do this? Can we scale it? Can we commercialize it? And these are big challenges in the area. What I'd say is, what I'm going to do is talk briefly about how we actually know these materials, where they are. We've done this, and sort of talk about this in a general sense. We've done this same type of strategy in targeting platinum-based drug, another cytotoxin with very narrow therapeutic index. This kind of carrier system is the kind of thing we design when we're interested in developing these. So we're saying, where are the materials? Where are the drugs? And so we can incorporate things like isotopic labels. Platinum as a drug, in this case, is very nice because there's not naturally incorporated platinum in biological systems, and so it's very easy to detect by a range of different methods. And then fluorescent dyes, this near-infrared dye, for example, that lets us do whole animal imaging of the kind I showed you on one of the first slides, but also high-resolution imaging like STORM, for example. And so just very briefly, we've been able to image particles as they enter cells through endosomal pathways. The particles themselves, in this case, when they get taken up by cells, are trapped within different compartments, but the drug leaks out. And we're able to track that using this kind of correlative or combined isotopic and optical nanoscopy or microscopy, in this case using something like NanoSIMS, which is secondary ion mass spec, or nano secondary ion mass spec, where we can see where the platinum is with respect to the carrier. And so you've got kind of a complicated system here, right? You've got a particle that's covalently bound to the drug, leaking the drug locally. And so you've got this burden on the characterization of the system in terms of tracking where the drug is compared to the carrier. And that's exactly what we've tried to achieve here. And I'll skip to this slide, just the top set of panels. This is now in the actual animal now. We previously published that in cells. Now we're looking at the accumulation of a platinum-based carrier within tumor tissue. And we can see here the N15 cyanide labeled signal. That's coming from our drug, intravenously injected, collecting within the tumor. And this overlays with the platinum-based drug, and then we can see the drug leak over time. So we get very local concept of how, a very local view of how the drug actually leaks from those aggregates as I described. And this is an ongoing, as I said, challenge for us and others in terms of tracking the carrier and the timing of this and the localization. As I said, what we're interested in here is not just tumor or cancer treatments, but also other diseases of inflammation. Now, the reality is when you work on xenografts like what we're working on here, these are human tumors in mice. So these are xenograft models where we've inserted a human tumor in an immune-compromised mouse. these tend to be leaky type tissues that are already predisposed to be capable of targeting nanoparticles. And so we've kind of stacked the deck in our favor when we use these types of models. The model itself is also at many times, especially for nanomedicine, not necessarily translatable to humans. So we got interested in other types of diseases, in particular heart disease, precisely for that reason. The models themselves more closely reflect human disease, and the human disease is untreatable at the sort of time points where we think nanomedicine has an influence. And I'll show you that in a second, but because they're exactly the same materials, I'm going to go to another challenge question for you about sort of the basic concept of how we believe these materials actually work.
For our next challenge question, it is, what is the major thermodynamic driving force for micelle formation in water from surfactant assembly? So I'm going to read through these and then I'm going to launch the poll question. Is it that attractive forces between the polar head group and the hydrophobic portion of the surfactant? Is it repulsive interactions within the core of the forming micelle between hydrophobic portions of the surfactant? Is it entropy associated with the desolivation of water from the hydrophobic portion of the surfactant as the micelle assembles? Or is it repulsive forces between the polar head group and the hydrophobic portion of the surfactant?
Correct answer is C and D. And so I mentioned the entropic effect of assembly, but also you have a separation between the polar head group and the hydrophobic portion, giving you the shape of that surfactant. Now, this is exactly the concept that we want to use in targeting the materials, because we want to change those interactions between the head group and the hydrophobic portion in order to ultimately change the shape of the assembly. And we use that same concept to go after this particular type of disease. In this case, as is shown on the slide, this is a collaboration between our group and Karen Chrisman. She's a bioengineer at UC San Diego and an expert in biomaterials for in the attempt to heal, in particular, MI, but also other diseases involving heart disease. You can see that this affects many, many people, and this is quite well known. In fact, a particular shocking statistic is many of the people, of course, many whom ultimately die from cardiovascular disease, die, in fact, from MI itself. And so you can see here a diagram of what happens to us over time, depending on how we eat and genetic predisposition. So we have a blockage that causes an ischemic event, so a reduction in blood flow to the heart, in this case at the left ventricle. So you have a blockage in the coronary artery. This blockage then becomes freed, and you get a reperfusion event, a pulse of blood into this location. And this sets off a set of events that results in inflammation, as you would normally have in any kind of healing tissue. Except this goes wrong, and of course this is in your heart, and so you can't put this in a sling as you would if you damaged your arm playing tennis. This continues to go on and beat. And as this occurs, inflammation spreads, as is shown in the diagram here. And in some cases, extraordinarily, within a very short period of time, your heart can enlarge and cease to be a pump. And of course, what we want to do is come in and intervene right at the beginning of this. We want to prevent as much cardiomyocyte death or muscle tissue death as we can. And we want to try to control this negative remodeling or this global remodeling process that ultimately destroys the heart tissue. And what we'd like to do is come in at that first step. We can't take any of these routes, right? We don't want to do it in surgery later on. We can't inject directly into the damaged portion of the heart. What we'd like to do is do an intravenous injection. Now, it turns out, as Karen pointed out to me early on, that the heart tissue right after infarction has the same sort of enzymes expressed locally. And so you have this pattern of enzyme expression that's exactly the same as what we see, or very similar to what we see in the inflamed tissue associated with metastatic cancer. Except in this case, it's more of a natural process. And what we do is we initiate these heart attacks in rats by blocking the coronary artery for some amount of time, like 30 seconds, and then release the blood again. So we replicate what's happening in people. And in fact, the progression of the disease then progresses in a very similar fashion. The tissue becomes very porous, so nanoparticles can get out of the vasculature into the muscle tissue, but they can come right back out. They come right back out of the muscle and into the bloodstream. And so we got interested in whether or not our sort of phase change could get a particle to go out of the vasculature and become physically trapped. And so again, these are the types of surfactants, again, assembled based on those principles that we just talked about with the challenge question. So hydrophobic region, hydrophilic region, hydrophobic region, hydrophilic region. In this case, we carry a tag and we can do this of various types. So we make a rhodamine-based tagged particle, for example. We inject intravenously, and we see it circulate and then accumulate over time. On this next slide, what you can see is a tissue slice from one of these rats. You see the left ventricle here. You see a very large site of heart attack. This is this white portion here where the muscle tissue has been obliterated. And in fact, white blood cells have invaded. The tissue is very porous, and so particles, if they were injected, would sort of get out of the vasculature, explore this space, and be pumped right back out. That's what I was talking about. Over here on the left-hand side, you see what healthy, this nice pink color in the H&E stain that we see here. And again, you see the scale bar. This is a rat heart. On the next slide, what you'll see is a little bit of a movie made out of the tissue slice. Now we've labeled those muscle cells in green, and we've labeled their nuclei in blue. And you can see the nice, healthy, tight tissue on the left-hand side there where there's no infarction. On the right-hand side, at the site of infarction, our particles carrying the rhodamine dye in the false color image here are red. You'll see us turn off the channels. The blue of the nuclei of the white blood cells turns off. And you see this really nice localization of our materials within the site of infarction. They've localized and stuck there because of the enzymes that are present, those same enzymes that we saw in the tumor case. So we'll zoom out here and I'll summarize this. So again, we've got this left ventricle. You've got this tissue here that's completely destroyed, so no muscle anymore. You've got this scar forming, and you've got it filled with white blood cells that are producing these enzymes. Our particles target to that location. Now, in this case, the particles are useless. They just carry a dye. But what we're doing now is developing systems where they could carry inflammatory or pro-inflammatory, anti-inflammatory type agents. They could carry peptides to help differentiate the muscle tissue to fill this void. And so we're now at the very beginning of looking at whether or not this is truly going to give us a good therapeutic strategy beyond just this imaging data that shows that this principle actually works. I'll skip this next slide in the interest of time and sum up. So what I've described to you, or I've hoped to describe to you, is what we've taken as a new approach to trying to trap materials within diseased tissue. We've used very similar signals that are associated with inflammation to do so. And we've basically used very simple polymer chemistry and the sort of basics of how you assemble particles of different shapes to build up these types of responsive systems and then tested them in animals. This leaves me with plenty of people to thank. I didn't do any of this work in the lab. My students shown here have worked on different aspects of this. I had other people's names on the slides, in particular Maria and Naneki work on both of the platinum-based systems. Chris Barback is a technician who's still at UCSD. He's an avid surfer and refused to move with me to Chicago. Cassie is a student who worked on the taxane delivery, so the Paclitaxel delivery that I described to begin with. I mentioned Karen Chrisman. We also have some clinical collaborators and some other people mentioned here who've helped with electron microscopy. This is my group now at Northwestern working on these types of projects and other aspects of this, and I'd like to hand over to the moderators.
Nathan, Andrea, thank you very much. Excellent. The questions have been coming in fast and furious here, so we're going to have a full question and answer session here, and I'll do my best. If you still have questions, please feel free to type them in, and I will try to handle them as best I can. Andrea, we're going to start with your presentation. This is a question from Stephanie. Is the shape changes that you showed in these hydrogels, is there any way to make this reversible? That is, can you go back, and have you thought about those kind of strategies?
Yes, that's a great question. And to an extent, they are reversible. So let's say we expose one side of the hydrogel to generate folding, and we get a tube. So using the simplest example, if we then exposed the other side in an equal manner, then it would counteract the original folding, so we can get it to go back to flat, as long as we can get the light in there. So it is possible to reverse the shape change, but there's a limited number of times you can do it. So then if we wanted to go back and expose one side, then we don't get as great of a gradient or a differential in the swelling. So there's a limited number of times you can go back and forth.
I see. Another question about the hydrogels with not with respect to going back and forth, but can you design things to have tunable degradation? I mean, we saw that the light affects the degradation, excuse me, the hydrolysis rate. So can we tune it such that you might consider like longer term implantable hydrogels, so you really don't want them to ultimately degrade? Can you say a word about the ultimate degradation or the ultimate fate of these? And this is a question from Shafak.
Generally speaking, our hydrogels are fairly stable unless you expose them to light. So within the body, they should have, I'll call it a reasonable lifetime, but we're using PEG diacrylate, whether or not the photodegradable group is incorporated. So we're limited by the lifetime of those, and they can undergo hydrolysis over the long term. There are other chemistries possible that our group and others have explored, where we don't have any hydrolytically degradable linkages within the hydrogels. So it's possible to make something essentially that only has these ether linkages and carbon-carbon bonds, as well as the photodegradable group, to prevent degradation wholly.
Okay, I see. And one more, this was from Mark. What about polyacrylamides? Is there any activity on polyacrylamides? Because I know they're used for kinds of hydrogels for other biomedical type applications. Is that something that is often done?
Yeah, so polyacrylamides are used obviously for analytical purposes like gel electrophoresis, and people also will modulate the mechanics of polyacrylamides to investigate this question of stiffness. We've actually been collaborating with someone else here at UCLA, where we've incorporated our photodegradable group into the polyacrylamides to make them tunable as well. So definitely another polymer that is widely used.
Great, thank you. Let's shift gears and go over to Nathan. So here's a question for you, Nathan. This is from Jennifer. You talk about the shape change mechanism, and kind of going from spheres to cylinders or something like that. But then you refer to kind of an aggregation phenomena. Do you feel like a precipitation-like event inside the tumor when your enzymes degrade the peptides, and it's actually just aggregation, they become no longer kind of micellar? Or how do you view that after the degradation by the enzyme takes place?
Yeah, it's a good question. So they do aggregate together. They go from a spherical structure to more of a network phase, though, which is still a micellar type phase. So what we believe happens based on our studies in vitro and actually in the animal as well, is that the particles don't just stick to each other and crash out, or the polymer just doesn't irreversibly precipitate. The polymer chains actually exchange between the polymers, and they undergo a true phase transition. So one of our pieces of evidence for that comes from some work we did really early on, where we made two identical particles. We actually made the polymers in solution, and then we split it into two pots. We're able to terminate the end of one of those polymers with a donor, a fluorescent donor, and the other polymer with a fluorescent acceptor. So we make two different particles, one that is the donor particle and one that's an acceptor. We intravenously inject both of them together at the same time, and then we get a fret signal from inside the tissue. And so this means that the dyes were in they're within tens of angstroms of each other. And so we can see that, yeah, it's like a true scrambling of the dye, and we've quantified that in vitro, of course. In vivo, it's a little hard to quantify, but we still see the effect. So one thing we can do, for example, is intravenously inject the donor labeled particle, and then wait a day or two, and then inject the acceptor, and we don't see the fret signal. They actually accumulate and aggregate separately from each other. So you don't get a true entanglement of the chains. They aggregate together. So if you look at light microscopy, you see them stuck in the same parts of the tissues, but the dyes aren't talking to each other because they're not close to each other enough. So that was a early on, yeah, big question for us, and I think it speaks to the big challenge in deciding and figuring out exactly what the nature of these materials is in the tissue, and whether or not they're a real network phase or a hydrogel kind of eludes us right now as to exactly how they look inside the tissue. But those are the types of pieces of evidence that we use to call it a phase change.
Thank you. There are a couple of questions, one by Alexandra and another here about the type of bonding that you used in your polymer. So you use ring opening metathesis polymerization, and you connect drugs and peptides through various bonding strategies. Can you talk a little bit about kind of hydrolytic stability of those, and as it relates to efficacy? That is, if the drug is, you know, really covalently bound to the backbone, does that make a big difference? And then the second part of that question is, are there other strategies out there that you might use besides ring opening metathesis polymerization?
The linkages that we used as I showed in the presentation, were all ester, so covalent linkages via ester bonds between the polymerizable group and the drug. We picked on a portion of the drug that's easily modifiable in that way. If you could do several different things with this. You can make carbonates, carbamates, esters. These are sort of known hydrolytically degradable bonds that are used. In fact, on Taxol itself, it already has some of these types of groups that are hydrolyzed in vivo. And so, whether or not, if you made this an irreversible linkage, we would expect it to have killed the drug completely. We haven't done that exact control with this system, but you could envision something that's slowly hydrolytic, like an amide, for example.
Right.
But yeah, that type of thing could be potentially tuned. And so you could make the core a little bit less hydrophobic and maybe have more access. What happens is, as you hydrolyze the drug, the core becomes hydrophilic. So you have a carboxylic acid, a carboxylate present now, which means that water can penetrate further and further. And so we believe the particles sort of unravel as you hydrolyze the core, and the polymer becomes hydrophilic. Now, the backbone is not degradable at all in this case, and that's where the part of the question about leading to another polymerization method comes in. And indeed, that's exactly what we're doing. ROMP turned out to be really good because you can polymerize all kinds of stuff. It's incredibly functional.
Very versatile. That's right. Yeah.
Very versatile and functional group tolerant when you use ruthenium, these types of initiators. But you end up with this polynobornate. It's kind of a weird polymer, and it's not degradable in any sort of time scale that would make sense. The good thing about it, what maybe serendipitously makes this work, is they do have high TG. So they're not cross-linked physically, but they're very stable upon injection. So these are stable down, sub-nanomolar, below what we can detect reasonably in terms of CMC. And so these are very stable, and then they're kind of spring-loaded because they're kinetically trapped. We are making particles now that have tunable TG and tunable degradability. We'll see how well they do, but we may have sort of stumbled upon maybe the best system by going after something because of the functional group tolerance. But yeah, we are definitely trying to do this with a degradable version. That's one of the big next steps for this.
Thank you. Andrea, we're going to come back to you in a couple of practical questions. One from Helen. I think you may have mentioned this earlier, but first of all, how do we measure the modulus of these hydrogels? What's your main tool for kind of estimating the modulus? Remember when the cells were growing on stiffer or softer parts of the hydrogel? And I guess you maybe mentioned this, there are strategies to increase the hydrogel modulus. I think you focus largely on decreasing the hydrogel modulus. Can you comment on that?
So for measuring the modulus, it depends. If we just have a large bulk sample, then we'll do what's easy, just a normal mechanical testing system. But for most of the examples where we're surface patterning that you saw today, we actually use AFM to measure the modulus. And we do this on fully hydrated samples, which is a bit of a tricky experiment, but my student is very, very talented. And the second question was about increasing the modulus. Certainly, there have been other people that have worked on that. You can go back and try to further cross-link the material. So if you have a photo cross-linkable material, sometimes people will go from one modulus to a stiffer modulus by an additional cross-linking step. Laura Suggs at UT Austin, I think, also reported using some ionic cross-linking to increase the modulus in a dynamic way. It's a little bit trickier because if you have an equilibrated sample, then the chains are farther apart and you're trying to bring them closer together, and that's a little bit more difficult to do than to just break chains. But there are definitely reports in the literature about that.
This is a practical question about manufacturing the gels in a sterile fashion. Is it important when you're doing these studies to, it's kind of a technical, practical question, important to do these in sterile environments, I guess? And how do you take precautions when doing these cell kind of studies?
It's definitely important. We don't want to grow mold or bacteria in our incubator and contaminate our samples. The easiest way to sterilize our samples is actually to sterile filter everything before polymerization, so before cross-linking, and then keep it in a sterile environment. Obviously, you can't autoclave these. You can't put them under UV lights because they'll degrade, etc. The other way we sometimes will sterilize them is we'll incubate. If we make the hydrogels before adding the cells, we'll incubate them in a 70% ethanol solution and then dilute that out with water and eventually media.
I see. Okay. And related to that, Ying Kai had a question about the UV light intensity. I guess this is also related to a question from Marta about actually doing these kinds of chemistries while things have been implanted. I guess this is tough for UV light penetration, but the real question is how about the UV light intensity on the viability of the cells just alone? I'm sure you do control experiments along those lines, and also the possibilities for doing this maybe kind of really in an in vivo situation.
Yeah, two great questions. So we've actually done a number of studies on the light intensity. The long story short is that at the intensities we work with and the exposure times, there's no effect on cell viability or gene expression, at least in human mesenchymal stem cells. So this was published in PLOS One a number of years ago. We undertook quite a big study with that. If you also go to the literature and in our own experience, the big danger with the wavelengths of lights that we work with and the intensities is really you have to be careful not to generate heat. That's what tends to result in killing cells. But these are pretty low level exposures. Getting it into the body.
Right. For example, doing this in vivo in a kind of, in situ kind of application.
Yeah, so we typically view it as more of an in vitro tool doing stuff in the lab versus stuff that's actually implanted. It is hypothetically or theoretically possible to do this in an implantable system. If you do something just under the surface of the skin, you can get UV light to penetrate maybe about 1 millimeter or a little bit less. We can think of topical applications. There's a lot of different lasers and light sources that have been developed for dermatology, and there are also laparoscopic delivery of light as well. So there are laser catheters and things like that if we really wanted to go in vivo as well. So there are possibilities, but I do think the practicality of it is this is, at least in my mind, more suited as a laboratory tool than something we will, or a topical approach versus something that we would implant long term.
Thank you. Nathan, we'll finish the question and answer session with you by just a couple questions. So one is kind of a general question. You've talked a lot about injection. Do you think some of the strategies that you're talking about could be applied to oral drug delivery? So taking a pill and getting through the intestinal wall, or is that kind of strategy focused more on injectable drugs?
In theory, it could be. These probably wouldn't work.
Okay.
But one of the ultimate goals, of course, is to get our oral bioavailability. And we have some other approaches for doing this with some similarly designed systems, but a slightly different type of polymer scaffold. But that is a very good question. And I think ultimately, I think everybody would agree that is a sort of a sought after route. Both, of course, whether it's the intravenous or the oral, these are all sort of ways of getting into the general system. And then you want to make use of some targeting mechanism to then get it to where it's going. And so, yeah, both of these are viable. These are designed for IV, but mostly just because I don't think we have any hope of going orally with these particular formulations. But that would be a fantastic ultimate goal.
And then the last question here is from Jessica, and Jessica asks, what about taking your system and designing it such that you could have two different size nanoparticles being injected at the same time? Could you pick cylindrical forming micelles or spherical forming micelles and kind of tune the original nanostructure size and shape to investigate the difference between really big micelles and really maybe differently shaped ones?
Yeah, so compare different morphologies in
For
the
example,
initial
right?
IV.
Yes.
It's a really good question. There's a slide on that deck which I think is available, slide 46 in the one I have, but I gave a summary of the doxil and abraxane, and then underneath that there's the 3 different references there.
Oh, good.
Disha and Mitra Gotri have both looked at this, and Samir in particular at UCSB has looked at the role of geometry of the materials, both in vitro and phagocytosis and sosis, and also by IV injection. We've done a small amount of work on it as well, looking at intraperitoneally trapping particles based on them being high aspect ratio versus being spheres. And we and others see various different types of effects depending on the length scale. And so very, very high aspect ratio particles, and Dennis showed this in that paper, that Nature Nanotech work, show very long circulation time versus spherical particles. And so there may be something to that. And whether you could couple that with then some change in the shape so that, because the high aspect ratio of these big fibers don't then get into the target location either. And so coupling sort of long circulation time in one phase with a phase change that then localizes the material was something that I think a lot of people have thought about. As far as I know, that hasn't been exactly implemented like that. And some of these outcomes are a little bit unpredictable. That would be an excellent opportunity given using these types of surfactant assemblies, because you can access these different phases and then directly compare them, even though the shell chemistry, the type of electrostatics and things would be potentially identical. So could you narrow it down to just a shape effect? That's another challenge in doing that. We haven't done it. It could be done. When it comes to size, I think it's a really great question. The heart tissue that I didn't mention heals up over 30 days. So the pores right after heart attack, the vasculature is quite porous to the tissue, and then it seals up. So over time, different size particles would be suited to actually getting through the closing pores. And so you might change the treatment regime based on particle size over time. That might be a neat academic trick. I don't know how translatable it would be, but we've never studied that exactly, but that might be an interesting opportunity for particle shape as well. Particle size.
Thanks to both Andrea and Nathan for just an outstanding webinar here, how to design better biomedicine. And I think we all learned how to do that in this webinar. And I won't go down all of the questions that are still out there. They're really comments. Thank you for this excellent and very informative webinar, and thank you for this excellent opportunity. So I think there's positive feedback in general from all of the audience members as well. I do want to remind everybody online that this is the third in the 2017 series. Our first two are actually hosted on our new Division of Polymer Chemistry YouTube site. So if this was interesting to you, you might find the Power of Plastics Polymerized Ionic Liquids webinar interesting, or the Design of Next Generation Sustainable Polymers interesting. Both of those webinars are actually available on our YouTube website. So Nathan and Andrea, again, thank you, and I'm going to turn it back over to Mike to wrap up the webinar.
I would like to thank all of you in the audience for joining us today, and we look forward to seeing you again next year when the series picks up again. And as Marcus said, there are many benefits to being a member of the Division of Polymer Chemistry, including access to technical program, discounts and workshops, and much more. Remember that the first year is free, and if you'd like to find out more, you can reach out to Kathy Mitchum at kathyl@vt.edu to find out more info.